So thank you very much to Ors and everyone involved in giving me an opportunity to talk to you a little bit today about, as the title says, a little bit about neutralizing antibodies to SARS-CoV-2 and prospective applications. I really enjoyed Ying Tam's talk that just ended. I think it serves as a good conduit to what I'm going to be talking about a little bit, and maybe you will also have questions after that relate to both talks. You saw a slide similar to this one from Ying just a second ago. It shows the devastating or certainly astounding number of people who have been infected with SARS-CoV-2 since the pandemic was recognized, so since January. And as you can tell from the shape of this curve, which is now approaching 30 million, is that we're still on a relatively upward trend in terms of the number of cases, and certainly not great. We can also look at the deaths due to COVID, and what we can see, what we can appreciate, is that we're now globally, at least, on a downward trend. So the number of people dying from COVID is declining. But if you look at this graph, you'll see that we truly, for all means and purposes, we're on a second sort of wave of break, and that the first wave was between the end of February and early June, and there was a bit of quietness in May and June. And then, as most of you know, things picked up again. So we're on a downward trend, which is obviously good. But I think, as Ying basically alluded to, the general feeling is that a vaccine will be needed to be able to bring this curve down completely and not have a third or maybe even fourth wave. In the absence of a vaccine, or until we have one or maybe even multiple vaccines, there's also the belief that therapeutic intervention will be key. And while there are now a few drugs that are being used for treating people afflicted with COVID-19, most biotech companies are looking at the use of antibodies to address this problem. So biologics to infuse into people. So this talk will sort of try to bridge both of those things. Talk a little bit about why antibodies are important and how they might be used. So what do we know truly about antibodies to COVID? And obviously, knowing what the antibodies look like and what they bind to can be informative to both vaccine design, if it ends up being required, and therapeutic application. And the most common antibody, or the antibodies that are the main focus of for application, are what are called neutralizing antibodies. And for those of you who don't know, this schematic illustrates what neutralizing antibodies are. So at the top of the slide is a diagram of a virus, obviously. And neutralizing antibodies are the antibodies that bind to the spike proteins that cover the surface of the virus, and that results in coating of the virus and preventing that virus particle from being able to infect its intended target. And so neutralization is basically the ability or the event that occurs when an antibody binds to a virus particle and neutralizes its infectivity. Neutralizing antibodies can also bind to virus antigens, including the envelope protein, when expressed on the surface of infected cells, and through Fc-mediated effector mechanisms can lead to the clearance of that cell. That obviously is also a mechanism that, or an activity that is mediated by CD8 T cells, but antibodies via their Fc have the ability to kill or mediate the killing of infected cells as well. So neutralizing antibodies can block virus directly, or they can block the spread of virus by killing infected cells. So what do we know about neutralizing antibodies in the case of SARS-CoV-2 infected patients, which is informative, arguably, for understanding what, for example, a vaccine would have to emulate or not emulate to be effective. And so if we look at this paper from Wang from just recently, as most papers these days, on the kinetics of neutralizing antibodies in SARS-CoV-2 infected patients, the nice thing about this study is it's one of the few where there's data on the development of neutralizing antibody responses in two populations, if you will, in two groups of patients. Those that are severely ill or were severely ill, and those that are mildly ill. And what you can appreciate, I hope, is first that neutralizing antibodies tend to be in COVID-2 infected individuals, tends to start by around day 10, so about a week into the infection. And in severely ill people, they climb very rapidly to very high titers, pretty much in everyone. If neutralizing antibodies don't develop by day 10, they certainly do by 2 weeks in. In comparison, and maybe strikingly, in mildly infected individuals, it appears that neutralizing antibodies might develop on average somewhat earlier, but they tend to not be as robust. And this goes back to the notion that those who are mildly infected or mildly ill, I should say, who maybe are even asymptomatic, don't develop very, very strong and robust neutralizing antibodies, and therefore may be at risk for reinfection. On the bottom of this image, you can also see that in this study, sera from these either severely ill people or mildly ill people were tested for neutralizing activity, also against SARS and MERS. And what you can see from this is that there's very little cross-reactivity, or there tends to be very little cross-reactivity between antibodies that are developing in SARS-CoV-2 and other coronaviruses that we've had experience with, SARS and MERS. Which of course says that if we develop a vaccine or antibodies against COVID-2, they likely, at least currently, very few have the ability to actually cross-neutralize, and presumably, if we had to experience another coronavirus, another novel coronavirus, there's likelihood that antibodies that are developed now may not be protective against something that may come in the future. So let me just go back to this. So on this x-axis, another thing that I should point out and that I didn't was that the time frame of studying these individuals was between the onset of illness and about a month and a half into disease, so day 45. What is not known, or what is poorly known, I should say, is the longevity of these antibodies. How long do they persist? Do they remain very high, which would offer immunity against reinfection, or do they decline? And most people think that they decline. So although neutralizing antibodies to SARS-CoV-2 do develop, they develop in people who may be asymptomatic or don't suffer very much from the illness, and certainly in people who develop very strong illness, there's a question, or there's some concern about the longevity of the immunity that those antibodies might be able to offer. And one of the primary reasons that there's that belief comes from what we know from SARS and MERS that have obviously come before. So this very elegant review by Stan Perlman shows that similar to SARS-CoV-2, people who several years ago infected with SARS or MERS who develop severe disease tend to make very robust neutralizing antibody responses, but there's a peak and then it basically settles down. More importantly, people who develop mild disease, at least in MERS, there's also a peak, but basically neutralizing antibody longevity basically wanes about a year into a year post-infection. And there's general belief that that may be the case for, or will be the case with SARS-CoV-2 as well. And so there's some concern, as to whether people who are infected now, whether they will be protected in the future, but it also has implications for vaccines, because the question is whether, how you might deploy a vaccine, or what the policy is going to be around vaccine implementation going forward, because a single immunization or even a series of immunizations may not be enough to keep the virus at bay, and that's certainly something that will need to be addressed in time. Additional support for the notion that neutralizing antibody titers will wane comes from this study from Otto Yang, where they looked at people who were suffering mildly from COVID-19, and basically the punchline is here, where it says that the half-life in people who were studied over a 90-day period, the half-life of the antibodies that they observed, neutralizing antibodies, were 36 days, right? Meaning essentially that there was a fairly rapid decline, and that these people most likely would not be protected several months down the road. So, neutralizing antibodies, but what I haven't said yet, at least I haven't said, is what a target for neutralizing antibodies are. But if you've heard Ian Tam's talk, you know, or if you've been following the news, you know that the target for neutralizing antibodies is this blue blob here, the spike protein, and as illustrated in one of the previous slides, that is the surface glycoprotein. The structure of it has solved. A recombinant form has been determined, and it's a trimeric structure. One of the promoters is shown here in this darker blue, and the two other promoters are shown in light blue. This purple thing here is the receptor binding domain that was mentioned in Ian Tam's talk, and that is the domain, the entity that the virus uses to attach its to ACE2 on its on target cells. And so antibodies that are developed, neutralizing antibodies, they target predominantly this spike protein, and many of them are targeted to this, what is called abbreviated the RBD of SARS-CoV-2. And this is just an illustration, an example of a paper from Marit Gill's group in the Netherlands, together with Roger Sanders. It's published in Science. And it shows, well, first of all, they took several patients from whom they isolated antibodies, and they could categorize these neutralizing antibodies into various groups. There are about 11 of them, and they call them clusters. And then they could basically determine by cryo-EM they determine the structures of these various antibodies in complex with a recombinant form of spike glycoprotein. So up here, or what you can basically see is that many of the antibodies, even from different clusters, they all basically target the RBD, albeit from different angles. So their epitopes all overlap, but there's different sort of fine specificities. But basically by the number, you can tell that the main target is the RBD, and this is why their vaccine approaches are focused on RBD. You can also appreciate this antibody over here from cluster 9, which binds to a non-RBD site. So there are epitopes, neutralizing epitopes on the S protein, additional to the RBD, but the predominant target is the RBD. I did want to take a few minutes to wrap up and speak about ongoing work in my lab, and that is looking at the glycan shield, what we call the glycan shield of CoV-2. So many enveloped viruses are expressed on their glycoprotein, glycans, obviously. Shown here is examples of HIV and flu, and these purple things here are the glycans. And what you can appreciate is that, for example, in HIV, it's heavily glycosylated, and that is, the virus does that to protect itself from neutralizing antibody responses. And so too, SARS-CoV-2 is heavily glycosylated, and shown here are the glycans in yellow and the RBD domains on each protomer colored in purple, this pink color, and blue, which sit at the top. We can also color the glycans according to the type of glycan that is present. And what I'll highlight here is that these orange and green glycans, which sit here at the bottom of the spike, and form a sort of ring around the bottom, presumably to protect the stalk region from neutralizing antibodies. With that, I will end. I will acknowledge the person in my lab who's been doing most of the heavy lifting on the work that we're doing currently on the glycan antibodies. That's Curtis. We've gotten some collaboration from Masa, who's a virologist here in the faculty. We're working with Jean-Philippe at the University of Toronto with his postdoc to determine the structure of these antibodies in complex with the spike glycoprotein. We've received antibodies, CoV-2 specific antibodies from Dennis Burton's lab and Devin Salk, and we're planning some in vitro neutralization experiments with live virus in collaboration with Martin Petrich at the BC CDC in Vancouver. Thank you very much, and I'm happy to answer any questions.
Thank you, Ralph. We are a little bit out of time. There is only one question which I will ask quickly, but I would like to encourage everybody else to continue to ask questions, and maybe Ralph could answer them and take advantage of the question and answer a virtual platform. What is cluster 9 neutralizing if it is not binding to the RBD? Steric hindrance?
Yes. It's assumed that the N-terminal domain is also, so it's presumed that, so what you saw in that picture is just a Fab arm of the antibody. That's done to facilitate the crystallization. But you have to imagine that the full antibody, when it's bound to the spike glycoprotein, would be 3 times much larger. So it would block or at least hinder the ability of the spike protein to get close to ACE2. Yes.
Well, thank you very much. And I would like to thank all of our speakers in this short session. Thank you.